No.
But virus cannot survive without a living thing. Virus survives in the cell of a living thing. This is the basic difference. This is the basic difference, and this is the cause why the treatment of virus is tough. Because it is inside the cell. Bacteria is not inside the cell. Bacteria is outside the cell, and from outside it destroys the cell. So we can destroy bacteria without destroying the cell. But antiviral disease, the antiviral drugs are difficult to handle because virus is inside the cell. This is also corona, but this is novel coronavirus 19 with more virulence. So due to some mutation, it has become so dangerous, and that's why the mortality and the morbidity is more.
Right. Now, I understand that it starts with concentration in the throat, and so when we sneeze or cough, billions of such particles are expelled and can be transmitted to another person.
Yes.
So is that the basic mode of transmission? So what about touch? So a little bit on this about the mode of transmission.
The basic mode of transmission is a droplet infection. It is an airborne disease. But if a person sneezes or cough, the range is more than 2 meters, and the millions of virus comes out with droplets. So it is a droplet infection basically. Nothing better than soap. And soap does not kill anything, neither bacteria nor virus. It simply reduces the surface tension, and due to reducing surface tension, the things which are which sticks on your hand or stuck in your hand, they are removed.
Namaskar. Welcome to this session of Straight Drive. As you just saw, in our last session 2 weeks back, we had a good interaction with Dr. Rajiv Srivastava on some basic COVID issues. We have invited Dr. Rajiv Srivastava once again to dilate upon some simple misconceptions. Please welcome Dr. Rajiv Srivastava.
Namaskar, I am Dr. Rajiv Srivastava.
So, Dr. Saab, with your permission, I will again start my session with you with quoting Shakespeare. In Twelfth Night, there is this famous quote which says, better be a witty fool than a foolish wit. Or in As You Like It, you have Touchstone saying that the fool doth think he is wise, but a wise man knows himself to be a fool. So we have all these foolish wise people who feel that the vaccine is just round the corner. And we have many fools, I would say, who think that the end of the world is about to come. What we need, in my opinion, is to be a wise fool and not a foolish wise person. That is, if we need hope, we need an informed hope.
Yes.
That means it should be based on reality, but hope is important, but we should avoid wistful and foolhardy hope. So in this part 2 session on COVID, my first question to you.
Yes.
So we know antigen. I think, as I understand, it's a molecule capable of stimulating an immune response.
Yes.
So, like our SARS-CoV-2, the virus that causes COVID-19, so has several antigens. At the same time, an antibody is a protein produced by the immune system in response to exposure to antigen, and it helps eliminate the virus.
Yes.
But of late, these two terms by common people like me are being used kind of interchangeably. So let's get this away first. Is in respect of testing, the antigen testing and antibody testing, and in respect of development of vaccine, please explain these terms.
First we come to antigen. The term antigen is used for any kind of pathogen which creates a disease in human body, like amoeba, bacteria, virus. These are known as antigen to body. Antigen enters from various routes, from drinking water, from air, directly from the blood, or as we know that in particularly AIDS, hepatitis B, they can also enter your body during a sexual act, and a blood transfusion or a injection which is contaminated with these things. Our enemy, I should say, enters in the body, then our body has some defense mechanism to deal with it. First, it sends its army in the form of white blood cells, but sometimes white blood cells are not enough, not very powerful army to deal with all antigens. Then what happens? Body creates antibodies against that particular antigen. There is a very complicated mechanism how it develops. I don't want to go in that detail because it's for professional purposes. We are telling our things to general public who wants to know what is antibody. Antibody is a type of protein. Several antibodies are formed. The first antibody which is usually body forms is immunoglobulin. We call it immunoglobulin. That is immunoglobulin M. M is formed first within 7, 8, 9, 10 days. Then after 25 days, immunoglobulin G is formed. Both are antibodies. They are like cage which catches the enemy or the prey, and that is very effective. But unfortunately, it takes time to develop. It may take 15 days, 20 days, and during that period, your only hope is your white blood cells and phagocyte and some killer cells. A small army, not very powerful army. So at that time, if the pathogen is very strong and very virulent, very dangerous, then patient is in a great risk. After formation of antibodies, it is highly improbable that something happens due to that antigen. Then if something happens, that is the aftermath. The injury which was made by the pathogen, then it is only the pathogen would not be there after that formation of antibody. So this is the antigen and antibody. I think everybody is clear. Antigen is our enemy, antibody is our great friend, our very strong army which is made by our body.
So very, very briefly,
yes,
in this explanation that you've given, so just clarify the antigen testing and antibody testing.
Antigen testing, we test the pathogen. Antigen we take from the blood, we take from a swab, we try to find out where and which enemy is it. I want to recognize it. First to find it, but problem is there. There is a problem with antigen test, that initially the number of antigen are very less. You take a sample of blood, not necessarily that you catch an enemy, because then the number of enemy is very less. Later on, all antigen multiplies, all bacteria, all amoeba, all virus, they multiply and they will become millions. Then it is very easy to recognize, but then it will become late. We want to recognize the disease in very early stage, and in very early stage to find out antigen is very, very tough. That's why a person who is suffering from any disease, suppose only 2 days have passed, and we take a swab from nose, from the throat, or a blood test, but we don't find it. This also happens with tuberculosis. In the very initial phase of tuberculosis, we won't get the tubercle bacilli as particularly known as AFB, acid fast bacilli. We don't get, and we see the sample under the microscope and we find nothing. This is the problem with the antigen test. Okay. So the more popular test is antibody test. If antibody is formed, and I have told you that one antibody is formed within 7-8 days, but even then for that antibody you have to wait for 7-8 days. Then people developed another thing, that is PCR, polymerase chain reaction. This test, what we do in many cases like tuberculosis and also in this corona, what it does, this test is very peculiar. It makes a photocopy, like there is one photograph, it makes photocopy of antigen. So suppose one antigen is there, that PCR will make it thousand copies, and so we are really able to catch the antigen in very early stage, where a pathogen or antigen is very less, but we artificially made its copy to catch it. That is PCR, it is an antigen test. An antibody test we cannot do initially. After that we'll do, and usually we do after corona is over and we want to know that whether antibody is formed, we are safe or not, and that person can also give his or her antibody to save another person. That is known as plasma transfusion, as you know.
Okay, thank you. So that clarifies it. Antigen testing is basically to determine if you have the virus or not.
What does that
Antibody
mean?
Is like
to recognize that our army is prepared.
Yeah, you had the virus and now you are.
And both are very specific. Antibody of corona is very specific for corona. Antibody for typhoid is very specific for typhoid. So we can recognize that this disease is.
Right. So now about the vaccine itself. So you've seen all these wide claims from varied sources that virus vaccine would be available by end of the year or early 2021 and so on. Well, as we understand, development of a vaccine is a long drawn process, more complicated than developing medication, because in case of medication you are giving it to somebody who's already suffering, and perhaps little bit of risk can be taken in that case. In case of vaccine, you are giving it to a healthy person. And even if there is 1% chance of some
1% chance is too much, perhaps.
Yeah, so of development of side effects, then it can cause more problems than the pandemic itself.
Yes.
So explain this, and what is your take on how much time will it take to really get the vaccine?
First, I want to tell you what a vaccine is. Vaccines are basically divided into two groups: a passive type of vaccine and an active vaccine. Actually, the vaccine should be active. Passive is just a makeshift arrangement. Passive vaccine, as I've told you, somebody has developed antibodies and we take that antibodies from that person and then give it to that ready-made antibodies to somebody else. But that is not very effective because the body itself didn't make it. Somebody has donated that plasma donation, as you know. They are passive immunization. Passive vaccine. Passive vaccines are not very popular, not very effective. So we have to develop on active vaccine. Active vaccines means you introduce something in the body which mimics like an enemy, but it is not enemy. So there are several ways. You do some half killing of the virus. You weaken the virus and give it to the body. Or you only give the outer coat of that virus, and the body thinks that enemy has come, and it starts its all process to develop an antibody. But what is the risk? You are giving part of a virus, you are thinking that it is, we have weakened it quite perfectly and we did not cause the disease. But it may happen that it may cause the disease in spite of you think. Because there are several tests. initially vaccine is tested on a vaccine. First we think that which type of vaccine we will going to make. With our past experience, should I introduce live attenuated, means Admara virus, or we are totally killed virus, or one recombinant vaccine that is only outer layer of the virus. First you decide, then you give to the animals, then you wait to develop antibodies and see if an animal should not die. This is the first stage. It will take 6 months, 7 months, 8 months. Then you are satisfied that now it is quite reliable to give it to the humans. Then you start human trial. Human trial, you start and wait to develop antibody, and not only to wait for antibody development, but you also see about the side effects. These are usually done in 3 stages. First stage, second stage, and third stage. Each stage will take 3 to 4 months, means 1 year. Usually we have passed that 6-8 months with animals, and now it is 1 year more. If anything happens in between and then one person dies after giving the vaccine, the most healthy persons, most healthy volunteers are taken for this thing, but it may happen. Then again we now we are in a fuss. We don't know how this person died. There is some defect in vaccine. Which defect? We stop at that level and we try to find out the defect. Yeah. It will take time. But suppose second is clear, then third is clear, then problem is production. A huge, large production for millions of people all over the world is not an easy job. So what happens? Minimum, usually it takes 2 to 2.5 years minimum. But suppose government wants that it should be done early, but we cannot change the protocol. Yeah. Yes, one thing we do, we may increase the number of volunteers, and volunteers take money for this purpose. For risking their life, they take money. You take healthy virus, and various types of races. Caucasian race will may respond differently. We can respond differently. Negroid persons can affect, black persons can react differently. So it is a huge task and a huge money is involved. We hope, as you have told, is there any hope? Yes, there is one hope which I want to emphasize. Why we could not develop the vaccine against AIDS and hepatitis C? We could not. We have a vaccine for hepatitis B, but not hepatitis C. We don't have vaccines for AIDS.
Common cold also?
Common cold. As a very good. Yeah. Best example has Mr. Mani as well given a common cold. Because these pathogens change their appearance, change their way very frequently, that is known as mutation. Hepatitis C shows such mutation. AIDS virus is very, very difficult due to many other reasons I don't want to discuss here. It will take much time and a complicated thing. But this coronavirus, it is quite dangerous, okay. But fortunately, it is not showing very large mutation. A very small mutation is occurring. That is our only hope, that we will be able to develop the vaccine. And what about time? As I have told you, it will take 2.5 years. Usually it takes. You may shorten it to 2 years. Already we have perhaps 1 year is going to. I think first case was in Wuhan in December.
December.
This December, this is 1 year. If somebody says that this is coming in 4 months, so that will be a world record. But I think at least 8, 9, 10 months it will take. And sometimes what they do, before the third trial is over, they start producing the vaccine. Suppose the trial is okay, then we will use the vaccine, otherwise destroy.
That way you are ready with the vaccine.
Ready with the vaccine, but there is a risk. That risk is financial, not our life. That vaccine will be useless. You will throw in the dustbin. But we hope that third trial will be okay, and during third trial we start to produce. Then you save at least 2-3 months. That is the hope.
Okay, so I'll convert what you've just said into statistical and engineering terms.
Yes.
So sometimes you have to do field trials of a product,
Yes.
then go for extended field trial and so on.
Yes.
So here it's possible, there are two things are possible. The extended trial of the vaccine,
Yes.
the sample size can be increased so much.
Increased.
By increasing the size, perhaps the time period can be reduced.
Yeah.
Perhaps. And number two, you can take the risk of manufacturing a large number of a vaccine which has gone to stage 3 of trial,
Yes.
and be ready that the day the trials are over, you are ready to market it and ship it.
Market immediately.
So that is our hope. But with all this, as I can see, if I have understood you correctly, middle of next year is the earliest I think we can hope that we'll have the vaccine. So that clarifies the position quite a lot. Thank you very much,
One
Dr.
thing I
Saab.
want to say.
Yeah, sure.
One thing more. As you know, the vaccine is developed just to give some half-killed virus. Suppose we use the mask and a very low dose of virus goes into you. It also acts like a vaccine, at least in the healthy people. So mask is very important. That's all.
Okay. Yeah, so thank you very much for joining us today. And as I said, to clear some popular misconceptions about COVID-19, and we all hope we'll have the vaccine soon. Viewers, I hope you liked our program and are a little wiser than you were earlier. We'll be back with another burning issue on Straight Drive next Thursday. Till then, please give us permission to leave. Thank you.